Thanks very much. So, it's a bit loud. I've got the fun bit today. I promise I'm not going to talk about cannabis, but I will talk about a whole lot of other things. And mostly I'm going to talk about research because that's where the Hughes Foundation has really, really helped us, and you guys have all helped us as well. And I want to talk a bit about how that help is turning into new information and potentially new treatments. So just want to acknowledge the traditional owners of the land that we're meeting on today and pay respects to their elders past, present and emerging. So I'm really lucky. I've got a great team and this is part of the team yesterday meeting up at John Hunter. And a lot of you guys will have met the nurses and be well aware of the great work that they do. I want to talk a bit about some of the rest of the team though that you probably don't see from day to day. So when we set up this centre, we wanted to find a way that we could get people to work together because brain cancer research is divided up into a whole lot of different groups. And so we thought the best way to do this might be to think about what the themes of research were and then talk through some of these things. So Kath Adams is hiding at the back on the Mark Hughes Foundation stand. Kath and I look after patient experience, which is really the clinical trials, our fantastic nursing team, and the sort of intersection between you guys and the centre. Next is translational neuroscience, and Professor Hubert Hondermark leads this with Chen Jan. So Hubert's a neuroscientist, and that means he works on how the brain functions and how that interacts with tumours. And what we're realising is that the tumour talks to the brain and the brain talks to the tumour. And so there's a whole lot of potential ways that we can interrupt this process. And that's the work they're doing. So we obviously need new treatments, and Paul Tooney and his team work in experimental therapeutics, and they're developing new drugs, new ways of approaching treating the cancer. Richard Lobb and his team are actually based at the University of Queensland, and I'll talk a little bit more, but they've developed a blood test which we published earlier this year. Saad Ramadan and his team work in imaging, so PET imaging, but more particularly MRI, and the Mark Hughes Foundation has very kindly co-funded a new machine at John Hunter, at the HMRI, which I'll talk about in a minute. And then Matt Dunn and his team in the paediatric area. So national collaborations are obviously really important to what we do, and our view is that we're really keen on a Team Australia approach. This is a complicated problem. We're not going to solve it just in Newcastle. We've got to reach out to the whole country. We really want to break down silos, but we want to work out who around the country is doing great research and how we can bring these groups together and what sort of help we'll bring them together. And we don't want to leave anyone behind. So with clinical trials and things, it's hard to open these trials everywhere in the country. We want to find ways that people could be involved. Sorry, I'm struggling a little bit with the looking backwards all the time, so apologies if the audio's going on and off a bit. This is also meaning that we can collaborate with groups like ABCARA, the Australian Brain Cancer Research Alliance, with Cogno doing clinical trials, and then our collaborators at QIMR and University of Queensland. We also collaborate nationally with Servia, with Genesis Care and a number of universities and other philanthropic groups, and I can see the BTAA people there. We do lots of work with Craig Cardinal and the group from BTAA. Okay, so the first thing I want to talk about is the importance of data. So it's really, really important for us to capture as much information from patients as we can. And there's been a really good way of doing this developed in collaboration with the WEHI, but we're a part of it in Newcastle. This now involves about 20 hospitals collecting data on patients as they go through their journey. And it's things like, date of diagnosis, the the diagnosis, when you started chemotherapy, when you had radiation, all these sort of things. And that then goes into a really big database, which is matched with death registries, and we can get really, really good survival data for a whole lot of things. And it's been incredibly useful. And it's starting to answer really useful, important clinical questions. So far we've got 10,300 patients data in this. And the type of questions that we're asking is, when a patient recurs with GBM and they have surgery or radiation or chemotherapy, what works better, who survives longer, how can we sort of make a decision? And we're finding some really interesting things. The survival data, for instance, suggests that it doesn't really matter which of the three, the survival's about the same. And there's lots of bias in that. It's not a proper clinical trial, but it's telling us what's happening in the real world, which is incredibly useful. And so this data then becomes available for researchers to ask their own questions about their data and then potentially about the whole country's data. The other thing that I think has been really, really critical to our development of research projects has been the brain cancer biobank. And I know that we've talked an awful lot about this in the past, and I know a lot of you guys have helped an awful lot. I think what we haven't talked about is what we've done with the samples that go into the biobank and how that's then really changed things. So it started in 2013, and it's really carefully managed by Dr. Cass Griffin, who is, without doubt, one of the country's best biobankers, and it's housed at HMRI. And it now holds more than 400 patient samples, and these are blood samples, tissue samples, and also a really special bunch of patients have made post-mortem donations, and I'll talk a little bit more about that. But I think this unique collection has really turbocharged a whole lot of work around the country and increasingly around the world. And I'm sorry I wasn't here at 9 o'clock because we were on a meeting with our colleagues in the US who are so excited about this collection and how it could help us answer some research questions that they've started a very tight collaboration. The other thing that the biobank has allowed is our team, particularly our team of younger researchers, to get moving really quickly. You can imagine if you're trying to set up a clinical trial and then get the material from the clinical trial and then do research on it, that can be a really, really long process. The great thing about the biobank is that we've already got 12 years of data, 12 years of samples, and so my team of younger researchers can get going straight away. Thanks to the support of you guys and the MHF, we've now got 47 early career researchers who are working at the centre. And this is from industry honours students through to postdocs. So a really, really great team. One of the practical examples that I want to spend a little bit of time on was developing a blood test for brain cancer. So a lot of you will be aware of prostate cancer and the fact that we've got a test called PSA, which is really helpful. So a patient can go to their doctor, have a PSA test, and you can see whether the blood test's gone up or down, and that really helps manage the disease. We haven't got that in brain cancer, as you well know. And the nuisance is that we have to rely on MRI scanning. And MRI scanners are big and expensive and are not all over the country. We're sort of lucky in Newcastle, but you can imagine that some of our patients from further out, particularly some of Tash's patients around Tamworth are travelling for a very long time. We hear about patients travelling for 6 hours to an MR and then 6 hours home again. So we set out trying to find if we could develop a blood test. And this work's being led by Dr. Richard Lobb, who's a very smart scientist at AIBN at University of Queensland. And we've developed technology that's based on a chip, and we take a very tiny amount of blood. We can actually do it with 0.5 ml of blood, run that through the chip. The chip pulls out all of these things that are floating around in the blood called EVs. And EVs are like little packages of messages that are floating around in the blood. We can then concentrate those, so it really is like finding a needle in a haystack and then concentrating all the needles, and then we can prove that these come from the brain. They're floating around in the blood, but they come from the brain. And then we can look at what's happening with the markers on the outside of these EVs. and they've got some really cool technology that does this. And we've got this working. We published it in January, and the test's looking really good. We need more data though, but this research wouldn't have been possible without the biobank. So by collecting blood samples from patients, putting that together with MRIs that we've collected as well, we were able to work out when the tumour was getting worse and what the blood tests were doing. Another example was a marker. I keep talking about prostate cancer, but we try and use things that we've used in other areas because biology only has a certain amount of ways of responding. And there's a thing called PSMA, or prostate-specific membrane antigen, and it turns out this hasn't got a very good name because it's actually in brain cancer too. And this work was led by Paul Tooney with Kelsey Madsen, one of our postdocs. And what we found is that PSMA actually is expressed extensively in brain cancer, but it's not like prostate cancer. The PSMA is not on the tumour cells, it's in the blood vessels around the tumour, so it's slightly different. So Kelsey went back and looked at where it was and showed us very clearly that this shows the blood vessels lighting up on the left-hand side, see the sort of orangey area, and then the PSMA seems to be just associated with the blood vessels. And when we looked at whether that affected survival, it didn't seem to in initial disease, but by looking at patients where we had recurrent samples, we could show that PSMA actually was a better prognostic factor. So the red graph there on the right-hand side shows that the survival is actually different and a bit better. So that was something we weren't expecting. Why are we excited about that? Well, we can actually image a cancer with PSMA. We can put a radioactive tag onto the end of the PSMA, and then we can inject that into the patient and then put the patient in the scanner. And you can see on the left-hand side, we get a scan of the whole brain, but we also get uptake. If you look at the picture in the middle there, we get uptake around the tumour, and that can be really helpful in helping us know where the tumour is and where it isn't, and can be useful in planning treatment. Another application that we're really interested in is a project being done by Becky Stratford, who's one of our fantastic PhD students. And she's looking at a molecule called P75, which is a neurotransmitter. And we're interested in this because we're sort of going backwards. We've got a drug that's been developed for other reasons, and we think it would be really interesting to have a look at whether this was expressed in brain tumours. And again, we found that there's lots of expression of this P75. So Becky's working through the process of, how commonly is this expressed? Is this something that we could use potentially in patients? And again, wouldn't have been possible unless we had a whole lot of tissue banked for her to look through. And the meeting that I was held up with this morning was looking at the cell of origin of glioblastoma. So there's a group based at Duke University in the US, and Duke's got the largest neuro-oncology unit in the US. And using our very unique collection in the biobank here, we're starting to get to the bottom of where the cancer is starting off, perhaps what might be causing it to start, and then how it might spread through the brain and how these processes work. So again, wouldn't have been able to be done without all the help from you guys and the biobank. So I wanted to also show you a picture of the new scanner that we've put in at HMRI, and I think it's probably the most colourful scanner in the world, and we're really, really pleased with this new technology. So this is a Siemens SEMA X MRI, and probably the best in the country at the moment. And this is Xiaomi putting a patient through that scanner. But this is giving us a whole lot of insights into what's going on in the tumour. And what we want to do now is start to collect patient data, so collect MRI scans as well. There's a huge amount of information that's in those scans, and we're probably not extracting all of that information at the moment because there's just too much there. But what I want to do is make sure that we don't throw that information away. And so by banking the images just the same way that we do with blood samples or with tissue samples, we can then develop a whole lot of ways of looking at that information. And there's a really good project being led by Nick Hughes in our team, and he's using AI to look through large numbers of these images and to get to the bottom of what more we can extract from that. And that's also been done in collaboration with the Walter and Eliza Hall Institute, or our partners at the Brain Cancer Centre. The other thing that we want to develop is a new type of technology. I might just spend a few minutes talking about this. So this platform is called Theranostics, and it basically relies on three things. Firstly, there's a target that you can find on a cancer cell, and we've been pretty good at identifying lots and lots of these over the last few years, and then developing a drug that binds onto that. So the idea is the target is expressed on the outside of the cell, we have a drug that binds onto it, and then there's a little bit of linkage chemistry, and we can put a radioactive isotope onto the other end of the drug. And the idea is that it floats around in your blood, you give it intravenously, floats around and then binds to the tumour, and then this radioactive part blows up and the whole lot gets destroyed. Now, that technology is again being used in prostate cancer, but there's a huge opportunity in brain cancer. The only problem at the moment is that the drug companies are not particularly interested because prostate cancer and breast cancer are much more common, brain cancer is much less common. So this is the sort of thing that we can do in a university because we're able to do this and a lot of other people aren't focusing on it. So we're hopeful that over the next few years we can develop a drug that will do this and then work out how we can best use it in patients. So Kath Adams reminded me that we're very keen to engage people in our research and engage them in a few different ways. One is that we're very keen to hear from you about what you think are the priorities, and then when we've got specific proposals, the government's very keen that we have input from patients and consumers, families, of what they see the challenges are and whether they think that this research would be helpful and move the field forward. So I've left a link there to the for centre membership, but again, Kath and Sally down the back can give you some information. Kath, sorry, it's hard to hear. Do you want to wait?
One of the things we realised is that, as Mark said this morning, the first step we need to do is get on the bus. But one of the things that we've been learning in research is we tend to get on the first bus we see and we get really excited and we drive it and we lead the way. And as patients, your bus is coming after that and you have to follow us. And sometimes some of the ways we take you are not the ways that you want to go. And so we want you to be involved right from the beginning to say, this is the sort of stuff we want to know. These are the things that we find acceptable. These are the things that are too hard. So the forms that we've got on the table up here are slightly different if you're a member of the centre. But if you'd actually like to inform us, if you'd like to be part of a group of patients that say, we want you to look at these things, we want you to research these things, we'd love you to come and put your details down on one of these forms. Or even if you just want to be on an email list to understand what we're doing at the centre, come down. You'll get the email list either way, because form will actually put you into the research. So come and sign up if you're interested. Thanks, Mark.
Yeah, thanks, Kath. That's good clarification. So sorry, it looks like I'm running a bit short of half an hour. Did anyone have any questions or things that?
Yeah, hi. I think you may have actually clarified and answered my question, but I'll ask it anyway, so it's in my mind. Just very recently, I told the oncologist that I would not be progressing on stroke 2. I will be using some alternate therapies for benzoyl and ivermectin, and I also said that I'm open to any research offers. I'm open to being tracked by a student. I'm open to have a protocol I'm using if it's any value at all to anybody. I feel this method sort of blank stare, so I didn't know whether it's too small a cohort or whether it's too far left of field. With reference to what we were just told, which may be what I'm looking for. Are you able to just comment on that, please?
Yeah, no.
I was offering to put data into the space, so there have been some research papers coming out recently on the efficacy and the synergy between the two drugs. And but I have since backtracked, and I'm going to commence onto the STOOD 2, and then the decision will be if I piggyback out with the other drugs as well.
Yeah, so look, my problem is that I'm married to a clinical pharmacologist, and so she spent her whole life developing drugs, and so we're constrained by what we've got evidence for, what we've got, solid, supported evidence, and then that evidence needs to be accepted by the government in terms of funding. So, there's a fairly long and complicated process. We're really keen to have any data people are happy to share with us. So, certainly keen to keep track of how you're going. We can't assign a student to individual patients because, they're science students, generally not medical. But there's certainly ways I think we can work together, and yeah, very keen to have a chat.
Earlier, Dr. Vine was saying how MRIs can be difficult to interpret what's happened after surgery or chemo or the bed. Would a PSMA PET be better or one of a big expense?
Yeah, no, that's a really good question. And we did a trial a few years ago looking at a PSMA PET versus MRI, and then another type of PET called a FET PET. And the accepted standard PET is probably using a thing called FET. But look, sometimes PSMA can be really helpful. I'm always keen that we think about why we're doing a test and what you're going to do with the answers once you get it. I think the tricky thing we have at the moment with MRI is when people are progressing and how we judge progression or not. And I think the more technology we can apply to that, the better. And hopefully the blood test will also help with that. But no, it's right. You're right. We need to think about other ways of looking at things. And PSMA PETs are quite readily available because they can be made. The radioisotope can be produced in Newcastle, whereas some of the other things have to be flown up from Melbourne. And they fly up every second. They make it every second Thursday and fly it up. And if anything happens, it gets offloaded off the plane or something. It's really difficult. So we need to find better ways of doing that locally.
Any other questions? Please put your hands together for Dave.